Hello, this is our final project for Nanoscale Microscopy 2019. My name is Artem.
My name is June.
And I'm Jesse.
Our topic is the utilization of bacteria to degrade styrofoam. Styrofoam, a polyestyrene, is one of the biggest problems in pollution, producing 2.3 billion kilograms of waste each year. It has many problems, including being non-biodegradable, being an oil byproduct, and producing CFCs, which degrades our ozone layer. Therefore, with the problem of styrofoam, we want to create a solution. In order to address the problem, we propose to bioengineer an enzyme into a bacteria, which makes it sustainable in the economical and environmental world.
So now we'll talk about two precedents similar to our experiment. The first one is about the Pseudomonas pudia bacteria. And this bacteria was found by Kevin O'Connor and his European colleagues. And it is essentially able to consume pure styrene oil, which is a remnant of superheated styrofoam, and it's also a toxic pollutant. So it's all able to do this once you use pyrolysis to the styrofoam. And it's also able to consume 80% of the styrene oil, and also along with other toxicants. But the reason why, although this may seem very effective, the reason why this is not practical is because you have to superheat this styrofoam. So if you have to put that much energy into the process, it's not energy efficient. Our next precedent is about the Audinella saccharinicis bacteria, and this was found in Japan near a recycling plant, and this bacteria is able to consume polyethylene terephthalate, also known as PET, and PET is basically the things that make plastic water bottles, and it's able to break it down using the PETase enzyme. Although this experiment was successful, it's not applicable to styrofoam because PET and styrofoam are two different types of plastic.
So, basing our proposal off of these two precedent researchers, we propose that we engineer bacteria that could actually degrade styrofoam into harmless materials. So, after doing some research, we figured out that mealworms actually created enzymes called Exigobacterium sp strain YT2 enzymes that actually degraded styrofoam and made them into harmless materials. So keeping that in mind, what we propose to do in our experiment is to take the gene that codes for this enzyme, this particular enzyme, and take it out from the genome of the mealworm by using restriction enzymes to the DNA, so we could cut the gene out and isolate the gene. After the gene is isolated, we propose that we use a cycle called a PCR, which stands for polymerase chain reaction cycle, which basically sort of makes a lot copies of the particular gene that we want, so that we could work with these specific genes. So there's 3 steps to the PCR cycle, which the first one is denaturization, which is basically heating up the gene and breaking the double helix bonds. After that is done, there's a second step called primer annealing, which is basically attaching primers to the 2 sides of the DNA. And the third and last step is elongation, which is basically putting DNA bases, attaching them to the primers, and copying the DNA. This cycle is repeated over and over again, and each time the cycle is completed, the number of the gene will exponentially grow.
So after we created all these DNA fragments, we now want to implement it and put it inside the bacteria. So in order to do this, we have to get a plasmid, which is a circular piece of DNA, and we would cut the plasmid using restriction enzymes. And now that since we have a gap within the plasmid, we're able to insert our fragment of DNA into the plasmid, and we seal it all up using DNA ligase.
So with the resulting plasmid, in order to put them into bacteria and actually use these plasmids, we do this technique called the heat shocking, which is basically forcing plasmids to enter the bacteria that we want, and sort of having the bacteria force the bacteria to incorporate the DNA in the plasmid into its genome. So after all that's said and done, with the bacteria, we could culture them in an environment that is styrofoam rich.
So now that we have our culture of bacteria, we now want to see if the bacteria is actually consuming the styrofoam and actually digesting it. So we're able to do this by using the fluorescence microscope. And basically, we're able to do this by first getting the GFP gene from a jellyfish and integrating it into the bacteria's genome. And once we do this, the bacteria will be able to light up. So now that it lights up, we can put it under a fluorescent microscope, and we're able to see if the bacteria is able to thrive or it perishes. The reason why we chose fluorescent microscopy is because compared to the other alternatives, it's much more practical. So for example, if we used electron microscopy, the electrons shooting from the electron microscope would damage our bacteria, which would cause unwanted effects. And if we use, say, scanning probe microscopy, then it wouldn't yield great results because the scanning probe microscopy will only sense for the surface of the bacteria, whereas we want the entire culture.
So during this experiment that we proposed, there's one major challenge that we might encounter. So that challenge is basically, even if we incorporate the gene that codes for the enzyme, which is called Exigobacterium sp strain YT2, that actually breaks down the styrofoam into the bacteria that we want, the bacteria still may not be able to create this enzyme because, like, the reason being is because bacteria cells are much less sophisticated than mealworm cells, which the enzyme originated from. Therefore, the bacteria itself may not physically be able to create these enzymes. So one possible way to solve this problem is by sort of researching this particular enzyme in depth. So looking at structure and maybe its critical points that actually does the work of breaking down the styrofoam. And after we identify that, we can maybe modify this enzyme that is compatible with the bacteria and put the enzyme in the bacteria. So if everything goes to plan and the experiment does succeed, one future direction we could take this experiment is by mass producing the resulting bacteria and sort of releasing them into environments around the world that are maybe very styrofoam polluted, and thus cleaning up the environment.
In a short summary, we want to take a gene from a mealworm, make an enzyme, and then bioengineer the enzyme into a bacteria. If we are successful, we will be both economically and environmentally sustainable.
All right, questions?
Wow, another great presentation. Thank you. Question: how long do you think, or how long do you think it will take to degrade the styrofoam?
From the research we've done with mealworm enzymes, it is said that it could sort of, in a day, it could degrade, I think, a pill's worth of styrofoam, and so it is still a significant amount if we put a lot of bacteria into that use of degrading the styrofoam. So, so I believe it's not that much, but it is significant enough to sort of degrade enough styrofoam to make a difference.
I want to point out that rather than using, because obviously we can use the mealworm bacteria that already has the enzyme to break it down, but the reason why we're not doing this is that it could potentially become, if we introduce it into new environments, it could become an invasive species and it could throw off balance the general ecosystem.
Very nice.
All right. If there's no more questions,
Yes.
I wanted to say this was very interesting, and you guys did an awesome job. I really like learning about it.
Thank you.
On or not? Okay, there we go. Okay, so my question is, do you know what the proteins actually degrade this styrofoam into, and like how good is that for the environment, or what would we do with that?
So, sort of clarify your question. Are you asking what protein actually does this work?
No,
Like, what's the byproduct?
what's the byproduct?
Oh, the byproduct of actually this enzyme degrading the styrofoam is we do still need to do more research on this, but yeah, I can't really tell you right now because yeah, we're not really sure.
I think it broke it down into carbon dioxide and other organic molecules. So it's basically generally safe for the environment.